Okay, so let's talk about neuromuscular junction, an NMJ. So a neuromuscular junction is exactly what it sounds like. It is the connection point between the nervous system and a skeletal muscle cell, okay? So neuromuscular junction. So again, in order for contraction to happen, so we need calcium, but calcium does not get released from the sarcoplasmic reticulum unless the muscle cell is stimulated by a motor neuron, okay, stimulated by the nervous system. So skeletal muscle fibers contract only when stimulated by a motor neuron. So this is a type of synapse, and we'll talk more about this in chapter 10 when we talk about the nervous system, but a synapse is a functional junction or a functional connection, not a physical connection. So if you look here, this is the axon of a motor neuron. Okay, so an axon is a structure on a neuron that goes and connects with either another neuron or a muscle or an effector. Okay, and this axon is going to tell the muscle to contract. Okay. But there is a space between the two, so they aren't physically touching. So this is a functional connection, not a physical connection. And the synapse is a site where an axon and muscle fiber meet. Okay? So components of the neuromuscular junction. Let's talk about this. So there's a motor neuron. Here's our axon from a motor neuron, and it comes down. And these are the little axon terminals. And these axon terminals are going to form the neuromuscular junction. So this is a neuromuscular junction right here. Here's our skeletal muscle cell. There's a nucleus, all the little mitochondria. And here's our myofibrils, made up of thick myosin, thin actin, and they're organized by repeating sarcomeres. Okay, so we have to have a motor neuron, a motor end plate. So the motor end plate so if you look, this is enlarged. And if you look, the sarcolemma, which is the plasma membrane of the skeletal muscle cell, is invaginated. If you look, it dips down, but then it's even more invaginated, okay? What does invaginations do? It increases the surface area. This invaginated portion of the sarcolemma, where the motor neuron meets, this is called the motor end plate, okay? This is the motor end plate. Why do we need increased surface area at the motor end plate? It's because there are lots of protein receptors. So we talked about, remember when we learned about the plasma membrane, and there are lots of proteins that are embedded in the plasma membrane. We talked about receptors as being one example. These receptors are going to be where the neurotransmitter binds. The more receptors we have, the more neurotransmitter that can bind, and that will ensure that an action potential is generated on the muscle plasma membrane, on that sarcolemma. Okay, so this is the motor end plate, highly invaginated, so you can have more receptors embedded in it. The synaptic cleft, this is specifically the space between the axon and the motor end plate. So this space right here is specifically called the synaptic cleft. Okay, so again, it's a functional connection, not a physical connection, and that space between the two is the synaptic cleft. If you look at this motor neuron, the axon of the motor neuron, you can see lots of mitochondria release. So the processes that happen in the axon terminal are really energy expensive, so you need lots of mitochondria for ATP production. You also see these little bitty vesicles, and lots of them. These vesicles, these are called synaptic vesicles because they are at the synapse. Synaptic vesicles, These contain, in this image, it contains a lot of these little blue dots. Those blue dots are representative of the neurotransmitter, okay? So synaptic vesicles are found in the axon terminal, and they contain the neurotransmitter. Neurotransmitters, this is a chemical messenger, okay? So a neurotransmitter is a chemical messenger that when released by the axon will go and diffuse across the synaptic cleft to the receptors that are located in the motor end plate, and that is how the axon is able to communicate to the motor end plate to cause an action potential, okay? So they communicate via a neurotransmitter, okay? And in the case of skeletal muscle contraction, the neurotransmitter that excites a skeletal muscle, okay, the neurotransmitter is called acetylcholine. And it is always acetylcholine when we're talking about skeletal muscle contraction, okay? So this motor neuron is going to send a signal down. That signal is going to cause these synaptic vesicles, remember exocytosis, right? These synaptic vesicles are going to undergo exocytosis and dump the acetylcholine, that neurotransmitter, into the synaptic cleft. That acetylcholine diffuses across and will bind to all of the receptors that are located in the motor end plate. And when acetylcholine binds to the receptors on the motor end plate, that is going to cause an action potential on the sarcolemma. Right? Okay, you with me? That action potential on the sarcolemma is going to go, so let's go back a couple of slides. That action potential is going to travel down the entire length of the sarcolemma. Remember these T-tubules? These T-tubules are invaginations of the sarcolemma. So when an action potential is sent along the length of the sarcolemma from the motor end plate, that action potential travels down those T-tubules, and because those T-tubules are sandwiched in between the cisternae of the sarcoplasmic reticulum, when there's an action potential here, that triggers the sarcoplasmic reticulum right next to it to dump calcium out, right? So an action potential triggers the sarcoplasmic reticulum to dump calcium out, and now that calcium can bind to troponin. When calcium is bound to troponin, troponin changes shape, pulls tropomyosin out of the way of the myosin binding sites on actin, and when tropomyosin is pulled out of the way, now these myosin heads can bind to actin. And when those myosin heads bind to actin, now we can have actin sliding past myosin, and we can shorten our sarcomeres. Okay? All right, that makes sense? Okay, listen to this as many times as you need to understand that. This, you should recognize this tissue back here. These are all skeletal muscle cells, skeletal muscle fibers, and you see striations, right? Those striations are due to the arrangement of actin and myosin. So because they are arranged in sarcomeres, these sarcomeres give that striated appearance, okay? These little guys, these are axons from motor neurons, and they come down, and this little axon terminal connects with a skeletal muscle cell, okay? This is a neuromuscular junction. So stimulus for contraction, acetylcholine, capital A, capital C, H. Okay, this is how you abbreviate acetylcholine. On the test, if you use abbreviations, they have to be the correct abbreviations. So if you want to shorten acetylcholine in your short answer response, you can say you can write it out like this. Remember, acetylcholine is the neurotransmitter that gets dumped into that synaptic cleft, okay? It's that chemical messenger that is going to bind to those receptors on the motor end plate, and it's going to tell the skeletal muscle cell to cause an action potential. So acetylcholine is the neurotransmitter. A nerve impulse causes the release, so remember, a nerve impulse causes the release of acetylcholine from those synaptic vesicles via exocytosis. Acetylcholine gets dumped into the synaptic cleft. It diffuses across the synaptic cleft, and it binds to acetylcholine receptors on the motor end plate. That motor end plate is highly invaginated because more surface area means more acetylcholine receptors that can be embedded in it. More acetylcholine receptors means more acetylcholine can bind, ensuring that an action potential gets sent down the sarcolemma. Acetylcholine causes changes in membrane permeability. So this is where I'm going to leave off of my video, and I want you to go and I want you to watch the Neuromuscular Junction video that I posted, because you can actually, it does a really excellent job of showing you how acetylcholine gets dumped into the synapse and how an action potential gets generated on the sarcolemma, okay? So I want you to watch that video. Once an action potential gets sent down the length of the sarcolemma, remember, it travels down not only the sarcolemma, but it travels down those T tubules, and having an action potential on this T tubule causes the sarcoplasmic reticulum right next to it to dump calcium out. Okay? Calcium gets dumped out from the sarcoplasmic reticulum. That calcium then binds to troponin. Troponin changes shape. Troponin is also bound to tropomyosin, so when it changes shape, it moves tropomyosin out of the way and exposes the myosin binding sites on actin. Once those binding sites are exposed, the myosin heads can come down and attach. And that's how we get muscle contraction, okay? So watch that video, that link that I posted that shows what happens at the neuromuscular junction, okay? Then what I want you to do is, after that, I want you to watch the video of cross bridge formation, because the cross bridge formation, in order for the thin filaments, so in order for actin to slide past myosin, let's go back to this picture right here, these myosin heads are going to have to attach and pull, detach, attach, pull, detach, attach, pull. And that cycle is going to need to happen over and over again in order to pull the thin actin filament in towards the midline, okay? So that attach, pull, detach, attach, pull, detach, that is called the cross bridge cycle, okay? And that second video that I posted, that second link, shows you how that happens over and over and over again, as long as calcium is present, okay? Excitation-Contraction Coupling, this refers to the fact that you have to have excitation first and then contraction, right? So you have to have excitation via a motor neuron, and then contraction can take place. So that's what excitation-contraction coupling is. Okay? So I want you to watch the Neuromuscular Junction video, and then the cross bridge cycle video, and then you can read through these PowerPoint slides, because those videos explain all of this. I just have these PowerPoint slides on so that you can read through the process if needed. But all of this cross-bridge cycle, this is explained, and you can visualize it on that video. You can see the myosin heads attaching, pulling, detaching, reattaching, pulling, detaching. So it's much, much easier to visualize with the video, with the animation, okay? But all these PowerPoint slides are explained in that video, and they are here if you need a typed out explanation. And then, so relaxation of a skeletal muscle. So after you watch those neuromuscular junction video and the cross bridge video, then you have to think like, okay, how does it stop? How does a muscle relax? Because your skeletal muscle cells aren't contracting nonstop. You have to have relaxation happening too. So skeletal muscle relaxation takes place when, so first, the neural stimulation stops. So that motor neuron stops sending action potentials, okay? And when that happens, no more acetylcholine is going to get released. But you still have acetylcholine present that was already previously dumped out into the synapse, right? So how do we, as long as acetylcholine is present, you can stop dumping more. But as long as acetylcholine is still in the synapse, you're going to have muscle contraction happening. So there is an enzyme called acetylcholine esterase. Okay? Acetylcholine esterase degrades, decomposes acetylcholine that remains in the synapse. Acetylcholine esterase is always present. Okay? So it's present, and it's going to be decomposing and degrading acetylcholine, okay? And the reason for that is because we don't want to have our skeletal muscles constantly contracting. That's tetanus, right? When it just contracts and then never relaxes. So muscle, when that acetylcholine rapidly degrades via the acetylcholine esterase enzyme, that muscle impulse stops. And so no more action potentials are sent along the length of the sarcolemma. And so that's going to stop dumping calcium from the sarcoplasmic reticulum. But we still have calcium present because calcium was previously dumped. So there's still lots of calcium floating around inside of the sarcoplasm. So the stimulus gets removed, and then the sarcoplasmic reticulum stops dumping more calcium. But we have to try to get rid of the calcium that already exists. We do that via calcium pumps. So remember, a pump is a protein that requires, it's a transmembrane protein, and it requires energy, right? So a pump requires ATP energy. So embedded in the sarcoplasmic reticulum, which is a membrane structure, embedded in the sarcoplasmic reticulum are calcium pumps. And what they do is they use ATP energy to pump calcium from the sarcoplasm that had been dumped previously back into the sarcoplasmic reticulum for storage. Okay? So calcium pumps pump calcium that was previously dumped back into the sarcoplasmic reticulum to be stored for later. We don't get rid of calcium. We don't degrade calcium. We just store it for the next muscle contraction. Okay? So the calcium pumps pump calcium back into the sarcoplasmic reticulum and store it for later. When that calcium gets pumped back into the sarcoplasmic reticulum, well then calcium is no longer going to be bound to troponin. And so when calcium is no longer bound to troponin, tropomyosin, go back to this picture, when calcium is no longer bound to troponin, well, it changes back to its original shape, and tropomyosin slides back over the binding sites on actin. Okay? So the troponin-tropomyosin complex covers the binding sites on actin, and those myosin heads can no longer bind. Myosin and actin binding sites are now prevented, and muscle fiber relaxes. Okay? So, ATP, let's talk about this. You will see in the cross-bridge cycle video that ATP is required. Like it is absolute. So not only is calcium necessary, but ATP is necessary. And this requires lots and lots of ATP. So skeletal muscle contraction requires ATP, and skeletal muscle relaxation requires ATP because this calcium pump that removes the calcium and stores it back in the sarcoplasmic reticulum, this pump requires ATP. So not only is it energy expensive for contraction, it's energy expensive for relaxation. So rigor mortis, if you've ever heard of rigor mortis, that is when a dead body is like the muscles contract or the muscles stay contracted and tightened, okay? Why does that happen? They're dead. Their cells aren't making any more ATP, right? Well, ATP is required for this, and you'll see that ATP is actually required for the myosin heads to detach, and ATP is required to pump calcium back. So what happens is when the recently deceased individual is no longer making ATP, myosin heads cannot detach from actin, and so they stay attached and the muscles stay contracted, right? So ATP is required not only for contraction, but also relaxation. So if you don't have ATP, you won't be able to, you'll get to a point where you won't be able to relax your muscles anymore because this cross bridge stays and can't detach. You'll see that in the video, okay? All right, so watch that neuromuscular junction video, watch that cross bridge cycle video, and your quiz will be over the lecture from the PowerPoint, as well as it may cover information from those two videos as well, okay? All right, good luck studying. Email me if you have any questions.